Thank you. So, for starting, I would like to thank you, the organizers, for inviting me to this very nice conference. And so this would correspond to the encoding of the information into the synaptic structure. And the storage would be achieved by, well, the stabilization of these changes. So the idea is that, as was in this citation from Kandel, the idea is that the physical correlate of persistence of the memory as behavioral expressed is essentially the persistence of the synaptic structure as produced during learning. Okay? Now, until, as I said at the beginning, until very recently, even if we had a lot of, how to say, logical and circumstantial evidence, so for instance, we know that synapse can be modified and this modification can persist for quite long times, at least if the synapses are in a slice and there is no activity. But recently we can actually observe at least partly the synaptic dynamics, and this is due to the fact that basically pyramidal neurons have some specific anatomical structure that are called spines, where most of the synaptic connection from other excitatory neurons are formed. So essentially, roughly this amounts to the fact that if you can see a spine, then that means that with very high probability there will be a synapse on this spine. Even better, the volume, the size of the spines gives you information about the synaptic efficacy. Here you can see an example where basically the spine volume is correlated with 3 different measures of synaptic efficacy. In this case, it's the number of vesicles on the presynaptic side. You can see there is a kind of a nice linear relationship. Here, the spine volume is correlated with the postsynaptic density area, number of kind of receptors that you have on the postsynaptic side. So again, measure of efficacy of the synapse. And this probably is the more direct evidence where essentially through an engaging experiment, you can see that essentially the response recorded at soma is basically proportional to the spine volume, okay? So all of this to say that if I see a spine, then there is a synapse, and how large is the spine will tell me something about how strong is the synapse, okay? So now we can go on and try to see if brain really conforms to this theory, and this is a classic experiment where basically I think it's mouse has to do a kind of complicated task, as you can see from the success rate. So he has to reach through some very narrow slide to some food pellets. And you can see, well, it's a very complicated task, but still you can see that the rat is learning, so it's improving the performance, and this performance increase and then stabilize over days. Okay, so what we should see now? Well, now, if we were to look in the regions that are involved in performing this task, we should find some reorganization of the synaptic structure. In our case, given that we're looking at spines, we should see some change in the statistics of the spine in this area. And in fact, this is essentially what we see. So this is during the training, and you can see that there are spine that disappears. This is a spine that was there on the first day and then disappeared. And at the same time, there are some spines that appear. So this corresponds to kind of roughly elimination of connection and formation of connection. You can do this more systematically, and then what you discover is that essentially as a result of the training, you will have a specific increase in the rate of spine formation as compared to the different, here you have a series of control conditions. Okay, so this is very nice. probably there are also the change in the volume. They just looked at the number of spines. Now, what's kind of interesting and not immediately clear within the theory that I summarized at the beginning is that, in fact, you have formation and elimination also during control condition. So, in fact, in a situation where there is no explicit learning. And to some extent, you can see that this rate of change is not dramatically different from the rate of change that you observe during learning, okay? And so this is what we called, well, Hennenfent called synaptic volatility. So you have a reorganization of the synaptic structure in absence of any explicit learning. So they went on with Simon and, in fact, characterized some of the statistical features of this synaptic volatility. Is basically the data set that they used. And this is kind of image that you can see, and you can see that there is a change from day to day. So in this case, there are 6 sessions every 4 days. And what you see here is essentially each black line represents a spine that was there, in this case, in the first session. And then in the next session, if the black line is still there, it means the synapse was still there, the spine was still there. And otherwise, it becomes white. So you can see this where the spine is present on the first day. And you see that a fraction of them is going disappearing. And at the same time, you have formation of spines. And this dynamics going on. So yeah, formation and fixation. Now, it's kind of interesting is that if you look at the number of image spine per neuron, well, this number is approximately constant across the session. So you have formation, elimination, but the number is roughly constant. So there is no net change in the number of spines. And they did also a lot of work and looked at the volume of the spines. As I said, the volume is a measure of synaptic efficacies. And they looked at so-called stable spine. So stable spine would be the spine that are there all along the sessions. And they looked at fluctuation in the volume. As you can see, there are in fact fluctuation in the volume. And that have some interesting property in the dynamics, but for our purposes here, the point is that this distribution of spine size on the neurons is in fact again approximately constant. So you have fluctuation, but the total distribution is again approximately constant across neurons and across time. Okay, so to summarize this, essentially these data suggest that these ongoing spine dynamics, again, as I said, which is not related to any explicit learning, preserves the statistical features of the circuit. Okay? But, in fact, if you think of this in terms of synapse, well, then the fine structure of the circuit, which is essentially where the memory are or should be, well, this is undergone dramatic change, as you can see here, because basically over 20 days, almost half of the synapses that were there are not there anymore. New ones have been formed, but the old ones have disappeared. And also the stable one, which means stable over 20 days, they have undergone large fluctuation in the volume. That means that they have undergone large fluctuation in the efficacy of this connection. Okay? And so, of course, now you can ask several questions, and a pretty obvious question is, well, what's the effect of this reorganization on the patterns of activity in the network? Because patterns of activity in the network represents, to some extent, the information which is encoded in the synaptic matrix. So how this is going to be affected by these dynamics? So to do this, what we did is, well, we tried to build what I call the biological unconstrained model network. So it's kind of a standard network. We have 80% excitatory neurons, 20% inhibitory neurons. They are spiking neurons. They are randomly connected with some probability. And the distribution of synaptic efficacies are taken to be log normal. And the E2E connectivity we extract from the spine data, and I will explain in a couple of slides how we did. So what biologically constrained means is that we try to take all these parameters from experimental studies. And in this case, we have basically all the probability of connection and the mean and the variance of the different synaptic population. So we use this number. And so essentially, we were left with the problem of setting the external inputs that go to this network. And we choose these inputs so as to reproduce the average firing rate that are observed in the cortex. In this specific case, this would be kind of 1 Hertz for the excitatory population and 5 Hertz for the inhibitory population. Apart from this external input, which is different to the population, all neurons have the same parameters, okay? This is important. This will become important in a moment. Okay, so how we use the spine data to simulate these effects of ongoing volatility. First, we check that, well, the activity produced in the network resembles somehow the activity that you observe in the cortex. And in fact, this is what happens. So this is a trace from a synthetic excitatory neuron. This is a trace from a synthetic inhibitory neuron. So blue will always be excitatory and red will always be inhibitory. And so as you can see, this mimics some basic features. So you have fluctuation and the membrane potential, and the spikes are irregular and they resemble Poisson process. And also because of this random connectivity that I told you about, in fact, what happens is that you have very broad distribution of firing rates in the network. They are essentially very well fitted by log normal distribution. This would be the black lines. Here you can see in the log scale, so they look pretty Gaussian. And this again is a characteristic of activity in the cortex, so this right skewed long tail distribution. Now, I want to spend just a second to point out, because I say that all the neurons are the same, so where this difference in activity is coming from? Well, this difference in activity is coming from the fact that neuron receives randomly different inputs. So some neuron will receive, for instance, more excitatory synapse as compared to the average, so they will fire at a little bit higher rate, or vice versa, they will receive more inhibitory connections, so they will be firing, and this produces this. So in a way, you can say that this distribution is a signature of the fine connectivity of the network, okay? Okay, so now how we used the spine to simulate the reorganization of the network. Well, we did something quite simple, and essentially to each synapse in the network, we attached a spine from the experiment, okay? And this long succession. So if the spine in the experiment disappeared, we removed the synapse from the network. If the spine doubled volume, we doubled the synaptic efficacy, and so on and so forth, okay? And here you can see a snapshot of 10 by 10 connectivity. So blue is 0 millivolt, so there is no connection. This is a sparse network, so most of the connections are not there. And the color code tells you the efficacy of this synapse. And just by looking, by eye, you can see that this connectivity is significantly changing across these 6 sessions, okay? So what about now the distribution of activity in the network? Now, in this case, as I said, this dynamics preserves the statistical feature of the connectivity, so we should not expect any change in the global distribution of activity. And quite reassuring, that's in fact what we find. Here you can see distribution in the excitatory population, inhibitory population, and they are basically constant across the session.
You measure in order to change it, what's the pattern?
So here we have one network, and the connectivity between the excitatory cell is generated from the data. That means to each synapse, we assign a spine from the experiment. And then we change this connectivity as the spines change in the corresponding session. Apart from this, we don't do anything. Okay, so this is ongoing activity. We change the matrix, and we just see how this ongoing activity changed. Is clear? Okay.
There is no dynamic in the synapse?
No, this is 6 network. There is no synapse, it's fixed. Okay? Now, as I said, now we should expect now, on the other hand, a big change in the specific pattern of activity, because this specific pattern of activity really depends on the fine structure. And this again is 20 excitatory neurons, 20 inhibitory neurons, and this is the firing rate. Okay? And now this is a little bit surprising because it's very hard to spot any significant difference in these patterns of activity. So you can see this neuron is firing pretty high rate, and he keeps firing pretty high rate. This is low rate, and it's going the same for inhibition. So this is kind of surprising. So we quantified this, and this is the result of the quantification. So what you see, this black curve here is basically the autocorrelation between the synaptic matrices across different sessions. So essentially, this tells you how similar they are. Okay? So 1, that means they are identical, and 0 means they are completely uncorrelated. And
Autocorrelation of this matrix of what?
yeah, how similar they are, the synaptic matrix.
So you have from moment to moment this matrix, and you do
Right, so I take first matrix and do the autocorrelation with the second day. This will be this point. Then third day with the first one, this will be this point. This will tell me how much this is changed as compared to that one. And you can see, in fact, that the similarity goes down almost to 2.5. So it's pretty big change, which is consistent with the data that I showed you at the beginning. But what you can see is that, in fact, the pattern of activity in the network is hardly changed. So the pattern of activity in the excitatory population, which is the one that undergoes the most dramatic change, basically the correlation is above 0.95. So essentially, it's the same pattern. And you can see that inhibition hardly cares about all these reorganization of the connectivity. Okay? Which is, well, to us was surprising. I don't know if for you it was surprising, but it's pretty surprising. And so we say, well, maybe, given that some structure anyway is preserved, this is not going to 0. Maybe it's this residual correlation that preserves 4 minutes?
so the blue in the end.
Well, I had questions. I have 4 minutes. Okay, so what we did is, in fact, given that we are in a simulation, we can just rewire all the connectivity. Okay, so what does this mean rewiring? I take a network, and then in this case E to E, and then I take another network, is all the same connectivity, apart the E to E connectivity, which is completely generated from scratch with the same statistics. Okay, so this would correspond to 0 on this axis. And as you can see, again, essentially there is, even if you rewire all the E to E connectivity, which is 50% of the synapse in the network, in fact, there is basically no change in the pattern of activity. So we said, okay, let's try E to I. So this will be excitation to inhibition, and you see a little bit more change, but actually not too much change. And, well, then we tried the inhibition, and we tried inhibition to excitation, and now you can see that there is a very dramatic change. In fact, even if this is a much small number of synapses compared to this, but if you rewire the inhibition, essentially the pattern of activity in the excitatory network is completely changed. And even more interesting in some sense is if you rewire inhibition to inhibition, basically you get a much significantly larger effect on the excitatory population than if you rewire directly the connectivity on the excitatory population. Okay? Well, I think I don't have to spend. The conclusion of this is pretty obvious, is that in a way the pattern of activity in the network is very robust to reorganization of the excitatory connectivity, and at the same time it's very sensitive to change in the inhibitory synapse. We developed a theory to understand this and to try to predict this change, and this you can see, so the simulation will be the black dots and the theory will be the bars. You can see we have a pretty good quantitative understanding of this phenomenon. Actually, it turns out that to understand this is very simple, and you can think about this in this way. So consider a neuron in this network, and is receiving some connectivity from excitatory and inhibitory neurons. And now you can ask, what is the condition for when I switch, for instance, these two synapses, for not to have a change? Well, of course, I will not have a change if W1 is the same as W2. Of course, I will not have a change if Ni1 is equal to Ni2. This would be the activity. So in general, in fact, I will not have a change if the product of these two terms is the same. Okay? So this suggests that the larger is the variance of this term, the larger will be the effect. Okay? And now we can go in the cortex and check the numbers. So we can compute the variance of this term in the excitatory population. This will be this term here. It's not important, the formula, it's just the variance of this input that will come from the excitatory population. And we can do the same for the inhibition on the excitation. We will get basically the same formula within exchanged. And now we can compute this number and take the ratio. And then we discover, in fact, that the variance that is coming from the excitation is kind of 20 times smaller than the variance that is coming from the inhibition. And this is why actually changing the excitation has very little effect as compared to changing the inhibition. And if you are very curious about how this comes about, well, the main difference is essentially in the width of the distribution and the average firing rate. So activity in the inhibitory population is much larger and broader than the activity in the inhibitory population. Okay? So now this, of course, raises a problem for learning, because I just have showed you that no matter what I do with the excitatory connectivity, there is no way to change the pattern of activity in the network, so how do I possibly learn anything? And so we went back to the data, and we noticed that, in fact, what happens during learning is that you have a net change in the number of spines. So remember, I showed you the ongoing, where the statistics is constant, so there is no net change. In this case, you see that the rate of elimination is constant. I mean, doesn't change as compared to the control, but this is increased. So that means that neurons in this population will tend to have, on average, more connection than before learning. Okay? So we tried to simulate this process. So what we did is, we take some baseline network, and then we add a given fraction of connections to each neuron in this network. Okay? And then we did just the same analysis as before, a little bit more. And I'm almost finished. And then we looked at what happened to the activity. And again, basically nothing happened. Distribution are the same, almost the same. And if you look at the pattern of firing rates, I will quantify this in a moment. If you look at the pattern of firing rates, well, they are very, very, very similar. Okay? We did this with all the neurons. Now, one possibility is, in fact, that not all the neurons have this increase, but just subpopulation of the neurons inside this network will increase, and the other will stay the same. So basically, we repeated the same manipulation, but we did this only for 20% of the neurons in the network. Okay? And when we did this, surprise, surprise, we get huge change in the pattern of activity, as you can see here. And you can see also on the distribution. And in fact, it turns out that this as a function of fraction of neurons. You can see, well, if you don't change anything, this will be very similar, actually will be exactly the same. And then as you increase the fraction of neurons, you will make the pattern of activity more and more different. And then eventually you start going back so that if you change to everybody, essentially you have a very, very small change. Okay? Now something I want you to pay a little bit of attention is the striking difference between these two figures. So this was the ongoing plasticity, where we had, in fact, huge change in the synaptic matrix, but basically no change in the pattern of activity. And this is kind of learning-like plasticity, where we had tiny change in the synaptic matrix and huge change in the pattern of activity. Okay? So how this comes about? And now we have to go back to the balanced state that David and Carl talked about in first day. And so if you remember, this just very quickly is that, well, you have this excitatory and inhibitory population, and in principle, the total input going to this population is very large, proportional to the number, I finished, it's proportional to the number of neurons. But dynamically, in fact, the activity in the network adjusts so that basically these terms becomes very small, where very small means becomes of the order of 1 over square root of n. Okay, so these, in fact, eventually are small terms in the sense they are within the dynamic range of the neurons. But what happens when you basically add connection to a subpopulation within this network? Well, then basically these two populations, so the population that has increased number of spines and the population that has the same number of spines as before, well, then they will receive two different inputs. Okay? And the point is that given that they receive the same inhibition, these two populations cannot be balanced at the same time, in fact, because the difference between their inputs is, in fact, of order square root of n. So it is a large difference. And what happens is that when you do this, in fact, basically you transiently unbalance the network, and the population that has received this dominates the excitation, and the other one essentially shuts down. And this is what happened here, in fact. And I finished. So basically, well, I've shown that if you have change that preserve the overall distribution of activity in the network, then you will have very little effect because activity is stabilized by the inhibition. And so in order to make, in this situation, in order to make big change in the activity, you have necessarily to modify the inhibition. That one way you can kind of affect the pattern of activity through excitatory plasticity is to change the overall distribution, basically to unbalance the network. And just one speculation is that this, in fact, could be a way in which information is embedded in the inhibitory network. So basically, in this way, this will be a signal for inhibitory plasticity to start compensating this imbalance, and so we can discuss this later. Sure. Thank you.
Thank you very much.
Thank you.
Thank you. I thank the organizers for inviting me to speak in the last moment. I'm Menahem Segal, not Yadin Dudai, as I said before. And because I was only asked on Sunday to speak on Monday, I took the liberty of presenting things that are not really related to the main topic of the meeting, of this session, but then I saw that Gianluigi was actually introducing my presentation. So I thank you, Gianluigi, for A, talking about spines, and B, talking about small networks, because this is all I'm very interested in. And the reason I picked up this subject is because this is theoreticians versus experimentalists, and we collaborate with theoreticians. So David Hoffman is here in the audience, right? Thank you. And I collaborate with Elisha Moses in physics, and he has a collaboration with a group in Paris. So it's a second-order collaboration that I have with with the French neuroscience, or French theoreticians. And we're going to talk about determinants of network activity. All this work has been done, that I'm going to present today, has been done in tissue culture neurons. So I may not convince you that we know how the brain works, and for sure I'm not going to, that doesn't work. For sure I'm not going to convince you that I have a cure for Alzheimer, but as you will see, this is fun. So this is a neuron in culture, has lots of these dendritic spines that we talked about. Most of these dendritic spines have presynaptic terminals on them, so it's a bona fide neuron. And when we look at them, we fill them up with a calcium dye, they fire spontaneously. They produce network bursts. So this is part of a large network, several thousands of neurons, and they fire together. So the questions that we ask are, A, what determines the network? What starts activation of the network burst? And what terminates activation of that network burst? These are some of the questions that we ask, and there are lots of questions that one can ask using these tissue culture situation. For example, proportion of excitatory to inhibitory neurons in a network, we can control this as well. So just to show you what we do, we fill up the cells, we go for each cell in the field, and we produce this network burst, record this network activity using MATLAB and whatnot to be able to record as many cells as we can, all the cells in the field of view. And the first question is, do they change with age? This is something that we can easily control. At 1 week, we already see, at 2 days in culture, we already see some spontaneous activity. They synchronize already at 7 days in culture. And we can study what is the effect of different drugs that manipulate activity, and I won't go into this right now. Spontaneous activity evolves already at 2 to 3 days in culture, as soon as the cells make connections, and it can go on for as long as we record. What's the effect of growing cells in different concentrations? That is relevant to similar questions that was asked before. We can grow them in very sparse conditions, medium and dense conditions. We know that the proportion of inhibitory neurons is about the same no matter how big is the network. We find that network bursts are much larger. Cells are far more synchronized when they are in sparse conditions than when they are in dense conditions. So the network burst would be there, but the pattern of activity, the synchronicity, would be very different. What we found, this is related to our really basic interest in dendritic spines, we find that the sparse network dendrites contain far fewer spines than those in the dense network. So fewer spines, we know when we look at synaptic markers like postsynaptic PSD95, we see in the dense network all the synapses are on dendritic spines. In the sparse network, we find PSD95 or synaptic markers on the dendritic shaft. What does it mean? If we look at connectivity between cells in the sparse and the dense network, when we stimulate one cell and record from the other one, we can get a very large response in the sparse network, and much smaller response in the dense network. And this is for excitatory, this is for inhibitory connectivity. So dense network have smaller synapses. They are synapses on the spines, and the connections are fairly weak. Sparse cultures have synapses on the shaft, and the connections are very strong. So this is contrary to what people think about synapses being on shaft and on spines. We know from our work that synapses are on the spines are far smaller than synapses on the shaft. We collaborated with Elisha Moses to find out if the network burst begins at a certain location, and if so, are there any properties to the neurons that generate the network burst. And what we did was, this is Elisha Moses' group, they grow neurons on a long line. So it's one-dimensional network, and we know that the fibers go along the network, and we can just measure activity in these three positions, beginning and end of this loop. And then by looking at the latency of the burst, network burst, we can determine that there is a high probability that network bursts will be generated in specific places. We went on with this to find that in those places where network bursts are generated, there are fewer inhibitory neurons. There's more excitation in these regions. But we want to find if there is any leader neuron in the population. So for this, we went, again, in collaboration with Elisha Moses' group. We went to multi-electrode array. So we put cells in culture on these multi-electrode arrays. So that was published recently, about 2 months ago. We look at as many cells as we can, electrode number from 1 to 25. We line them up. We find, and this is illustration of activity, we find that under normal conditions, all the neurons would fire in bursts, synchronized bursts, right? Now, we go on, remove calcium. When we remove calcium from the medium, connectivity is lost. There is no synaptic connectivity, yet the neurons fire. So there is a lot of activity. If you summarize the activity here and here, it's about the same. But it's not synchronized as it is in the connected network. When we zoom in on the disconnected network, we can find that each neuron has a rhythmic activity irrespective of any other members in the group, because it's not connected. So they do have synchronous activity, and each of them can be described by power spectrum autocorrelation to find out what are the dominant frequency that they like to fire in. And surprisingly, the dominant frequency that the network burst occurs is the average of the individual network neurons. So the conclusion from this is that there is a democracy. The majority of the neurons, even though they fire irrespective of each other, when they are connected, the network burst is the average of the total activity in the neuron. What we can conclude is that when we remove calcium, excitability goes up. We know that from other studies that we have done and others. When we add calcium, synchronicity dominates, and excitability is going down. So here is an illustration of a network that fires spontaneously. Each neuron fires spontaneously in absence of extracellular calcium. You add 100 micromolar up to 500 micromolar, and the entire network synchronizes. And it synchronizes at the same rate that the average individual neurons is doing it. So that is fine, but can we determine if there is any leader neuron among the population? For this, we switch to a small network. It's more fun, because here you can look at the entire members in the population. If you want to do modeling, this is what we would like to offer you. So we can look at all the cells in the network. We can record from one or several of them. This is an illustration of one cell that is filled up with biocytin, and the other neurons are all in the network. We can look at the activity of these cells and begin to ask questions like, for example, how many cells do you need to have a network activity, right? Very simple. These are two cells, island of two cells. They fire in total synchronicity continuously. They do not need more than two neurons to talk to each other to be synchronous. Now you can take a network like this, mark with synaptophysin the number of terminals. Each of these is a terminal, and count how many cells you have, how many terminals you have, how can you characterize the network activity given the number of spikes, given the number of synapses and the number of, then this is a very tentative beginning of this kind of analysis. We know that the number of cells in the island is proportional to the number of synapses that you can have. This is pretty trivial. We did not analyze the activity yet, whether it is dependent or not. So for those of us who are interested in dendritic spines, what I can say is that this is a normal control culture, dissociated culture, all networks. So all these synapses are on the dendritic spine, as the textbook tells us. In the island, there are very few spines, if at all. But when you record activity of them, miniature excitatory postsynaptic currents, they are far larger than in the control case. So they make synapses on the dendritic shaft, and they produce larger synaptic currents than in the control. Again, contrary to what commonly is believed. We go on. We have another 10 minutes, that's okay. Again, we look at one of these culture, island culture, 7 to 10 cells can synchronize. We can record all of them. They are totally disconnected from other cells in other islands. And we can ask our critical question. When we have this spontaneous network activity, is there a leader? And you decide that this is the leader by looking at who fires first. You look at calcium imaging. The resolution is less than 1 millisecond. You can really tell which is the leader neuron. So here is one illustration of a small network with 7 or 8 cells, and they fire synchronously, except for one who doesn't like to fire, but you know, it's always you have an outlier in the group. You kill the first cell. So this is the cell that you thought is the leader because it just comes a millisecond before the other one. You kill him, and the rest of the network can still synchronize. You kill the second one, you just shove an electrode into the cell, you kill him, and the network is still synchronized. You kill the third one, and it's still synchronized. You are left with 5 or 6 cells. Now they are not synchronized. Each cell can still fire, but they are no longer synchronized. So the conclusion from this experiment is that leaders are not irreplaceable. For those of us who are worried about BB, this is the solution. So last issue is what terminates the burst, and this is work that we've done with David Hoffman. What we can do is activate by depolarizing current pulse neurons in the network, small network. You depolarize the cell, and you get a large burst that outlasts the depolarization pulse. Right? So depolarization produces a large burst of activity, which is a network activity. You wait another 5-10 seconds, and you give another burst, and this one is gone. You no longer get a full-blown network burst, but much smaller one. You need to wait. This is not just elimination. You need to wait 30 seconds or so. You depolarize again, and you get a full-blown burst. So the burst has some time course. So you need to wait for the burst for time to go by so that you can regenerate the network burst. We believe that this is related to the ability of the cell to release neurotransmitter from the terminals, and the reason for this is that if we change the amount of calcium in the recording medium, so again, this is one burst coming up a few seconds later, a much smaller burst, again, one burst, and much smaller burst. If we reduce calcium, reduce probability of release, we get the burst, and 5 seconds later, we get the similar burst. So we eliminated that duration-dependent change in burst frequency, indicating that this has to do with calcium, indicating that this has to do with release of neurotransmitter from terminals. We can cause, using latrotoxin, can cause a tremendous increase in spontaneous activity of the culture, but when we come later, we lose this time-dependent change in burst duration, and alpha-latrotoxin causes a tremendous increase in release of asynchronized release of neurotransmitter. And finally, this I actually took from a recent paper by David, came out last year, right? Work that we have done 5 years ago finally came out to tell us that this behavior that we have seen before can be explained by a facilitation depression model, and David can elaborate on this a little later. I still have 5 more minutes, but I give time for the general discussion. So the conclusion is that synchronized network activity is triggered in defined zone. We've seen it in the long line. The tentative chains can be found, and network bursts are generated by a majority. So there is no need for a leader neuron to, maybe there are leader neurons, but you do not really need them to generate network bursts in a small network. And that summarizes my presentation. Thank you. So just a brief introduction to the general discussion. We know that in different brain areas, the turnover rate of spines is different. We know that in hippocampus, turnover rate of spines is much faster than that in the motor cortex, for example. So if we are to use the same rules, why is it that spine turnover rate is different in different brain areas? Most likely it has to do with different molecules in the spines in the different areas. Neuromodulation is most likely also very different between different brain areas. We know that norepinephrine, we know that serotonin, acetylcholine, and dopamine are differentially distributed in different brain areas. So most likely they have a different role in long-term memory. There's a question of memory store, and we know that there are the extreme case. I don't know how many of you know the story of Pike. There's a person in the 19th century that could remember thousands of books. He would read a book in half a day and memorize all the. So he has a huge capacity for memory for the same size of brain that you and I have. But why is it that I do not remember my credit card number and he could remember millions of sentences? This is the question is, where is this memory? Where is the capacity? We know that, for example, blind people have better memory than regular people. There is the issue of reconstruction versus reproduction. This issue was brought by you. And the question is, when you memorize, do you reconstruct the memory or you strengthen it, reproduce it? And there is a big issue that came out in the past 10 years. Is the network dependent on activity, on morphology, or on some specific molecules? There was a suggestion that PKM zeta, suggested 10 years ago by Todd Sacktor. You block this molecule, you erase memory. That made a huge wave in New York because he promised that if you have a bad memory, I'll give you zip and will erase your memory. Is this real or not? Is this have to do with how longevity of memory or not? So these are the issues. Some of the issues, there are millions of other issues, but we have only about 25 minutes to deal with them. And please, could any of you comment on this or on any other issues related to longevity of memory, rules for memory, and whatnot?
I suggest just one that I want to come to this exceptional memory phenotype. So we had the case of Pike. There is a very famous case also in, I think the first described was by Luria. He has a very little book where he describes this person that could basically remember almost anything, and there is, I think, a fictional character from a Borges novel also, that Funes el Memorioso. And well, this of course they are kind of exceptional case, but I think they raise a very interesting issue, because one idea about why we forget stuff is that we have interference with other memories. So in a way we have some limited capacity and we have to make room for other stuff. Now I think that these kind of cases tend to suggest that maybe that's not why we forget stuff, because apparently there is much room, much more than we use every day. And I don't know, I want to know what do you think about this, if it's kind of makes sense to you. In sense, recently there is this idea that in fact also forgetting is a process which is actively controlled. And so in a way this kind of observation about exceptional memory, you think that they are kind of supportive of these ideas, so that forgetting is kind of controlled, or you are happy with the standard interference theory?
There are, yeah, I mean, they talk about memory in general without discriminating between different types of memory. I think it's problematic, especially if you talk about mechanisms. Now, some people have problems with memorizing faces. They don't have any problem with recognizing faces, but they have problems with memorizing faces, some with memorizing names, some have problems with memorizing places or texts, and they have very good other types of memory. So I think that even when you are talking about episodic memory and so on, you have to discriminate. I mean, it's not the capacity, for sure, it's not the capacity. The problem is probably something in the mechanisms which sits in the specific brain areas and take care of the memory in these areas, which is probably specific to some kind of entity and then some people may have problems with specific memories while having very good memory to other things.
I
any other comment on this? For sure, we know that there are different types of memory. Walking memory, very different types, and different longevity for each type. And most likely, people who suffer from memory problems have very brain-specific or location-specific disturbance of memory. So that may not affect other functions, but for sure, we cannot really talk about general rules when you want to compare motor learning that lasts forever compared to active memory or to other types of memory that may last a day or two.
Yeah, but even memory for faces, even episodic memory, to different things may be different, located in different areas, influenced by different processes, enhanced by specific neuromodulators and so on. So it's not very easy to generalize.
That's exactly what I'm saying too. I think that building a model for learning, a general model, it may not be that productive because it depends on the type of memory, the brain area. You may have different molecules involved in each of them. So developing a general model, as you showed in almost the first slide and the last slide, now this is what memory is all about, is not very productive. And the question is really, how do we get input from theoreticians, if we do, to direct our work? Because you can do millions of different experiments. You really want to come with an idea so that you can direct your experiment to test specific types of memory.
Yeah, it's about the different mechanism. So you talked about plasticity of synaptic inputs, but we know that there is also plasticity of intrinsic excitability of neurons. And recent work shows that when you can manipulate this excitability, you can also change memory and you can change the size of the engram and so on and so forth. So it is something that you also start to implement in your models and in your thinking. How do you see this dialogue between intrinsic plasticity and synaptic plasticity?
That's very good. Actually, this is a question to ask the theoreticians, because we know that dendritic excitability is modified by learning, is modified by LTP. We know that A current goes down so as to increase excitability in dendrites. So this is really not related to dendritic spines at all. This is in dendritic excitability. So whoever is doing a model should implement this change in excitability for sure.
So, sorry, probably, yeah, before I...
So I wanted to add on this. This is clearly there are a lot of changes, not only synaptic changes, and especially as you say, there is a lot of evidence that neuronal plasticity is accompanied by synaptic plasticity or preceded and so on. Now, I would say, this is my opinion, in the direction of going more close to biology, it's maybe interesting issue to try to implement this. Now, from a theoretical point of view, this change at single cell level, in fact, they expect to have a very small impact on the capacity of the network, in the sense how many memories you can store. And I would say that's why most theoreticians don't really care about single cell change, because this is not going to affect dramatically the network capacity.
So but now you have experiment that shows that actually changes the memory. So if you increase excitability by instance by increasing current in some cells, then you will change the memory of a given.
So I mean, now there is some new experiments that are going on where people try to change this level of intrinsic excitability, and indeed they find some effect on memory and also on the number of cells that are recruited to.
Right.
So I mean, even if on theory you think it doesn't have a strong impact, I mean, experimentally it shows that it matters, right?
So this is what's been shown, that in a way, because one question is, when you get some novel information, how do you allocate new neurons to this? Now there is some line of evidence that shows that, in fact, by controlling the excitability, so in a way, the more excitable in this specific moment will be the one that will encode. But this, let's say, classically in memory, this would correspond to the encoding phase. And this maybe facilitates the encoding or select by some process. But then there is the storage phase, right? And when you store memories, kind of a parameter which is important is given number of neurons and a given number of synapses, how many memories I can store in this network. And basically this modulation of excitability will not affect this parameter, but maybe will affect, well, initial encoding and probably recall of the memory.
I just wanted to comment earlier that I think there should be a distinction between task-specific synaptic plasticity and general homeostatic mechanisms, because, I mean, you said that maybe in different brain areas you would have different synaptic plasticity rules, but I think that some components would be different, but some components of the synaptic plasticity rules might be the same. Because you showed in your work, for instance, some gain control mechanisms, like if you have the sparse cultures, then they have the larger synapses, the larger ZPSCs. These are things that which I also see in my models. In models that kind of try to better represent information, when you sensory deprive the network, the synapses become stronger, the network tries to compensate in order to be more sensitive to the input. So it's kind of, it emerges naturally, these kind of things. So you can get them in models, and I believe that these kind of things, they happen in many brain areas, but they're not the only thing. And the harder thing for me as a theorist is to, to produce indeed the task-specific synaptic plasticity. It's highly non-trivial. I mean, to teach networks to do complicated tasks. But the homeostatic mechanisms, they are there, and that was the comment, yeah.
So just to comment on this, I think that you're right. We know that there are differences between different brain areas in different types of plasticity. Even in culture, we find differences between hippocampal and cortical culture. In homeostatic plasticity, we did not find homeostatic plasticity very much in hippocampus. We find a lot more of this in cortex. And when we looked back in the original studies of Torrigiano, they did it in cortex. Okay, so there are differences there that are seen even in the totally artificial condition, right? Now, another example is spike time independent plasticity. Everybody is now waving the spike STDP. And for those of us who have done experiments, not theory, experiments on spike time independent plasticity, find it very hard to reproduce the spike time independent plasticity, meaning that sometimes if the spike precedes or follows the EPSP, it may either increase or decrease it. It's not a general rule for this. Now, it may have to do with the tissue that you're working with. I know that the original observations by Markram were done in cortical slices, and the original observation in culture by Mu-Ming Pu was done in hippocampal culture. We tried to reproduce the hippocampal culture experiment. We could not find it. But, we did not use exactly the same. So this is really not a universal mechanism. Sometimes you find it, sometimes you do not. And it can go on for different, other different, what is considered memory or learning mechanisms or learning rules. It's highly tissue-specific, experiment-specific, and I should only add to this that, as you know, there were some studies by drug companies found that they were unable to replicate so many experiments, up to 70%, they say. Amgen did a study on this, were unable to replicate. So if you do not find, you do not use exactly the same condition that the original paper suggests, and you are unable to find it, you're not going to go to publish irreproducible results, you know. But if you read the literature, you think, this is it. This is a mechanism that if we stick to it, we can explain everything. No. Yeah, David.
Yeah, so are we allowed to ask questions about your talks?
Oh, me?
Yeah, both of you.
Yeah, okay.
So first of all, I would like to start with this bursting time. This is something actually interesting. This is something robust. The network show more or less the same bursting time with some variation. So there is something here intrinsic to a network, not to a single neuron.
That's right.
So, and it apparently doesn't need any kind of plasticity. I mean, it happened spontaneously, correct?
That's right.
So now, as a function of time, when the culture is aging, is this bursting time changing?
This is very interesting. No, we don't know that. We did not study it systematically.
Okay.
But for sure, it depends on the number of the cells in the network. It depends on a number of other things. But as far as age, we did not study.
Okay, second question. This is about the first part of your talk of the synchronization versus desynchronization with calcium. So do you think here that calcium, I mean, what he's really doing at the level of a single synapse would be like to play the role of, I mean, it is involved in facilitation. And so the facilitation is the reason why you have everything in synchrony when you have calcium, because when you have a spontaneous spike, then let's say at one neuron, then it will affect the ensemble, and the facilitation is the time to trigger everybody to fire.
Yes. So what, I mean, the situation is, as you described, when we remove calcium from the medium, this is what I did for single cells, removing calcium reduces the threshold for spiking. Adding calcium increases the threshold by up to 5 to 10 millivolts. So that's a very strong difference that is dependent on presence of calcium. It's got nothing to do with synapses. Just the threshold for producing a spike is going up when you add calcium and going down minus 50 millivolt to minus 42, 43 millivolt when you add calcium. So that by itself is a property of the spiking, not of the network, nor of the synapse.
Okay, and now I have a comment or question for Gianluigi. The idea of using empirical data from the spine and putting this in the model, I found it very interesting. Now, it turned out that looking at the volume of the spine is one thing, but what determines actually the electrical property of the spine is mostly the neck length, and not much of the head. And you can have a difference of potential of, 20, 30 millivolts. So the question is, of course, I understand, looking at information statistics about the neck is very different rather than looking at the statistics about the head. So this is one thing that maybe you want to think about. And the second thing is that not all spine are connected, as Menahem showed, only a few percent. So, this is also something to think about. Maybe it's 10, 20%. Menahem knows better than me.
Oh, well, yeah, of course, yes. That's true that the mapping between spine volume to synaptic efficacy is not as simple as I showed. clearly about connectivity in, I'm not an expert, but about connectivity in the cortex, they say 80–90% of the spines that are there, in fact, they have functional synapse. So this is true. I think that for the kind of argument that we wanted to make, the specific mapping between any characteristic of the spine and the activity, I think it's quantitatively important, but from a qualitative point of view, this would not really change. The point is there is some reorganization of this connectivity through change, could be change in spine neck, so in principle could be even larger than just counting the spine. What we show is that this can be pretty massive, and still, if the inhibition is stable and in a way the activity in the inhibitory population is larger than the activity in the excited population, which what seems to happen in the cortex, then this change will have little effect. And that's a way to, for us, that's a possibility how inhibition could contribute to stabilize representation or response of the network, even if you have this volatility that's going on. And the functional role of volatility is not clear, for instance. I mean,
just to comment on this, this is a technical issue. You cannot really resolve spine neck width in regular light microscopy, only in super resolution microscopy, and very few people have this ability. The one technical note. So you look at the spine size, because this is something that you can easily measure. The length. Length is another thing that you can measure, yes, of course. But there is another issue which is related to this. The illustrations that Gianluigi wrote were of spines every 4 or 5 micron. Now, in cortex, in the real cells, you have a spine every micron, sometimes even more than a spine every micron. So one wonders if you select the dendrites that have very few spines to have a nice illustration, or this is really some sick dendrites that are far away that have very few spines on them. Usually, standard spine density is about 1 spine per micron, not 1 spine per 5 micron. So that's a technicality that has maybe to do something with the interpretation of the results.
Yeah, so I had a question for both of you, actually, on the type of network. So the role of inhibition is central, and I mean, you know the paper of Misha Tsodik's 97 in the stabilization of networks with inhibition. And interestingly, Ken Miller has used this idea to show that in the visual cortex, actually inhibition can be used as stabilization of the network. So it seems that it's quite an important fact that inhibition could be there to actually stabilize memory and this kind of thing. So I would like to have your opinion whether it's linked to the work that Misha did in 97, and for Menahem, in your study, in your culture cells, is there inhibition? This is a question. And the second thing is, how does this relate to the work of Paolo Bonifazi and your ex-PhD student, Miri Goldin, that they did here in Lumini, where they found hub cells in hippocampal neurons, which were dramatically efficient. They could look with two-photon imaging at the network, the spontaneous network, and identify, this was the work of Paolo, which cells were much more efficient than the others, decide that this is the hub cells, and Miri was patching it, and if she's depolarizing, activating this cell, the whole network would go crazy.
So I think I would say no, they are not directly related, in the sense we're talking two different kind of stability. There is stabilization of the activity, of neuronal activity. In this case, I meant more as a structural stabilization, in the sense that you can have a reorganization of excitatory connectivity, but provided the inhibition is stable, or more stable than the excitation, this still would preserve the patterns of activity in the network. So I would say no, they are not directly related in any case. So
far as the inhibition, as I said, we can easily control them, the proportion of inhibitory neurons. Normally, we have in the culture about 20 to 30% of inhibitory neurons. They take part. We can see that activity is drastically changing when we block inhibition, so that's for sure. We have a simple trick to increase the number of inhibitory neurons. We can mix cultures from the hippocampus, which has, let's say, 20%, with cultures coming from striatum, 95% GABAergic neurons. So they like each other in the culture. We can look at activity changes under this. We've done some preliminary experiment, but not followed up on this. The second question about Miri Goldin, my former student, I should not comment on work on my former students.
A question to you. You showed that there is a change in the spine while learning, and you can reverse the situation, if I understand. Usually people talk about extinction. You don't reverse the situation. You learn something else that reverses the situation, but the spine does not return to be as it was before. Can you comment about it?
Well, there are some experiments that shows that if you do this reverse learning, well, you don't get exactly the same spines, but you get spines in same neurons, in the same structure. So, if I understand your question, right? You say do for some learning, then I do extinction, and what they saw is that the neurons that increased their number of spines, then actually they are the neurons that now decrease. So there is this reversibility of learning. So maybe something that I didn't mention is that this change in the number of spines, in fact, is only transient, in the sense that then they will go back to the same number. So this will equilibrate again. So eventually there is no net increase in a sense. It's just transiently. And that's why we believe it's important that inhibition also has to change, because if inhibition doesn't change, this just amounts to rewiring to some extent. And we show that rewiring has no too much effect.
In the pre-Botstinger complex, you have an ensemble of about hundreds of neurons that are also giving a synchrony, and when you kill neurons, you find finally that there is no leader. So what basically you have found in culture is something that is probably generic to some system that can generate oscillation without leaders.
If there are no further questions, you are released to lunch. Are there any announcements?